Hello and hi everyone, we are Group 7. We want to present about Enterobacter aerogenes. First of all, we go to introduction.
Enterobacter is a genus of a common gram-negative, facultative anaerobic, rod-shaped, and non-spore-forming bacteria belonging to the family Enterobacteriaceae. Two of its well-known species, Enterobacter aerogenes and Enterobacter croce, have taken on clinical significance as opportunistic bacteria and have emerged as nosocomial pathogens from intensive care patients pathogenic, especially to those who are on mechanical ventilation. Enterobacter aerogenes was originally named Aerobacter aerogenes and was later included in the genus Enterobacter in 1960. In 1971, this species was proposed to be renamed Klebsiella mobilis due to its mortality conferred by periticious flagella and its genetic relatedness to Klebsiella genus. It is interesting to note that phenotypic dissimilarities between Enterobacter aerogenes and the genus Klebsiella include not only the motility, but also the presence of ornithine decarboxylase ODC activity and the lack of urease activity in the Enterobacter aerogenes.
Recently, the whole genome sequencing of multidrug-resistant MDR clinical isolate, including cholesterol, suggested a possible reclassification of the species in the genes Klebsiella under the name K. aeromobilis. Enterobacter aerogene particular phenotype can be attributed to the horizontal equation of additional genes from other Enterobacteriaceae species and mobile elements that rapidly integrated and translated as easily as its own integrated heritage. For example, the particular genus and its assembly system have been acquired in block from the Sorrento genus. Plasmid conjugation is a chimera of transport and genetic elements conjugation integration of various bacterial aerogenes. Enterobacter aerogen can also contain 8 RNA operands and 87 RNA associated with the ability to translate imported genes from use different codons, improving its assembly to use integrated foreign genus. Enterobacter aerogenes have been involved to significant European outbreak between 1993 and 2003, and it's considered as the paradigm of opportunistic bacteria.
Okay, next we go to biochemical pathway of application.
Example for biochemical pathway of application is the use of Enterobacter aerogenes for co-fermentation of carbon sources to enhance the production of bioethanol. Enterobacter aerogenes APCC29007 is a gram-negative microorganism and can grow very well in both aerobic and anaerobic conditions in a medium containing a range of carbon sources such as glucose, glycerol, mannitol, sucrose, and fructose. Researchers investigate the optimal growth rate of Enterobacter aerogenes APCC29007 by using carbon sources and mixtures containing glycerol. Results shows improved growth rate and bioethanol production compared to glycerol-only mediums. These experiments demonstrated the co-fermentation of glycerol with various carbon sources by Enterobacter aerogenes APCC29007 to enhance bioethanol production. The co-fermentation of mannitol with glycerol was the best combination for the cell growth and production of bioethanol.
Okay, next we go to process. First process is optimization of hydrogen. Hydrogen offers excellent development possibilities as a concentrated energy source with high added value, particularly in sustainable microbial production replacing traditional fossil fuels. This work offers a fresh approach to investigating microbial hydrogen generation using combinatorial gene manipulation in the future. Second process is fermentative pathway of glycerol. This work explores the use of genetic engineering to improve bioethanol production. There was a huge increase in ethanol production. These findings could be useful in further developing bioethanol generators.
Next, the benefit of Enterobacter aerogenes. Firstly is probiotic in the digestive tracts of both human and animals, Enterobacter aerogenes and other Enterobacter species might have a probiotic effect. Probiotics are living organisms, preparations that enhance the microbial balance in the gut and benefit the host. Secondly, Enterobacter aerugine is versatile bacterial pathogen confronting antibiotic treatment. Over the past 30 years, Enterobacter aerugine has been identified in hospital ward as a significant opportunistic and multi-resistant bacterial pathogen for humans. The spread of Enterobacter aerugine is linked to the expression of detoxifying enzyme involved in the breakdown and inactivation of antibiotics as well as the existence of redundant regulatory cascade that effectively regulate membrane permeability, guaranteeing bacterial protection. Lastly, Enterobacter aerugine as a facultative anaerobe. Enterobacter aerugine can produce hydrogen at a higher conversion rate in strictly anaerobic conditions and exhibits strong environmental adaptability. Enterobacter aerugine also benefit from matured genetic modification technique, quick growth, broad substrate utilization range, and easy of cultivation. As such, it is regarded as the perfect stain for industrial-scale biological hydrogen production. Nevertheless, the actual production of hydrogen is far less than the theoretical 4 mol H2 per mol glucose due to the incomplete metabolic process. Therefore, to produce hydrogen, it is essential to comprehend and optimize Enterobacter aerugine metabolic network.
Last but not least, we have case study and published paper in Natural Resources field.
There are several cases exist regarding Enterobacter aerogenes, which commonly infect the respiratory tract, urinary tract, and even the central nervous system. But the case that I want to mention is newly reported in 2020, which is the infection of the lumbar spine. In this case, a 69-year-old female patient with osteoporosis who lately was diagnosed with Klebsiella aerogenes infection of the lumbar spine. For your information, Klebsiella aerogenes is the new name for Enterobacter aerogenes. The patient was hospitalized on 30 July 2020 with recurrent fever without any cause for 2 weeks and continuous lower back pain for 3 days. After analysis with the patient blood sample, K aerogenes was detected by blood culture and metagenomic next generation sequencing. Since Klebsiella aerogenes is resistant to antibiotic, intensive treatment and precise medication were done until the lumbar degeneration was improved. And the figure below is the article related to the case of lumbar spine.
Enterobacter aerogenes has been studied for its potential in hydrogen production through a process called biological hydrogen production or fermentative hydrogen production. This is a fascinating area of research with potential applications in renewable energy. For benefits of using Enterobacter aerogenes in hydrogen production, first, efficient hydrogen production. Enterobacter aerogenes is selected for its ability to efficiently convert glycerol into hydrogen. The optimizations of immobilizations techniques aims to enhance this efficiency. Second, continuous hydrogen production. The use of immobilized Enterobacter aerogenes allows for continuous hydrogen production. The continuous process is advantageous for maintaining a steady and reliable output of hydrogen. Thirdly, cell density. Immobilizations of the mechanism is designed to increase cell density in the bioreactor. Higher cell density is considered a prerequisite for achieving a high volumetric hydrogen yield. Last, adaptability to glycerol. While Enterobacter aerogenes is known for the capability to convert glucose into hydrogen, the study suggests that the mechanism can be adapted to utilize glycerol. Immobilization is seen as a way to enhance its tolerance to glycerol, facilitating the use of glycerol as a feedstock. For environmental sustainability is renewable research. Utilizing biodiesel byproduct as a substrate makes the process more sustainable and reduce reliance on fossil fuels. Nextly, reduce greenhouse gas emissions. Hydrogen produced through dark fermentations has the potential to be a clean fuel, as it does not emit carbon dioxide during production.
In the field of microbiology, incubator aerogenes emerges as a microbial powerhouse, captivating scientists with its potential application in various fields. Bacteria such as incubator aerogenes are important in biotechnology application. At the same time, these microorganisms have an extraordinary ability to produce enzymes, organic compounds, and other valuable substances. Incubator aerogenes has multiple functions in various industries, such as contributing to the production of important components in pharmaceutical, improving food processing, and advancing the development of sustainable biofuel useful for humans. Additionally, certain strains of incubator aerogenes exert good prebiotic potential. This beneficial microorganism, when consumed as sufficient amount, promote healthy good flora, digestion, strengthen the immune system, and may help preventing gastrointestinal problems. To understand the complex characteristic and genetic makeup of incubator aerogenes is important for humans. This knowledge fuels research and development, paving the way for innovative technology and medical discovery in various fields.